And so, as usual, I changed the title. Genome Editing to Cure SCIDX1. So a couple things here. First of all, I don't really represent Stanford University. I'm an associate professor there. If Stanford University thought I was representing them, I think they would fire me. And the second thing is I feel many of you who grew up in the U.S. probably watched Sesame Street when you were growing up, and there was that regular skit about one thing's not like the other. And I feel a little bit like that today, listening to all these companies present. I'm really going to be presenting our work in my academic lab that relates to a specific project. And of course, I think it's a real opportunity to do this here where there's so many companies and possibly as a way of partnering. I will disclose, as I'm required to by Stanford, that I have an academic founder of CRISPR Therapeutics, which is one of the 3 genome editing companies founded around the CRISPR technology. And so I consult for them, and just note that they're hiring if you're interested. So as many of you know, there are thousands of diseases caused by single mutations in the genome. So in the top left, there's a picture of a sickle cell, and as you know, that's caused by a single A to T mutation. And and if both copies contain the T at position 6, you have sickle cell anemia. Down in the lower left is a picture of a little baby boy in a bubble who had bubble boy disease, or SCIDX1. And unlike sickle cell anemia, this disease is caused by mutations scattered throughout the gene, leading to an inability to develop T cells and NK cells. So the point being is that we have a 6 billion character genome. I always make the point that while we talk about the human genome being 3 billion, as far as I know, I'm a diploid. And so I think we have 6 billion characters. And that's a 1.1 million page book. And there's a single typographical error. And the idea is that with genome editing, we can correct these typographical errors. So as I mentioned before, there are tens of thousands of such or probably over 10,000 such diseases, and I've listed a few here. And through this conference, you've heard a lot of different approaches to approach some of these diseases. I will, in particular, be focusing on SCIDX1, which falls in the category of primary immunodeficiencies. And the approach that we've been developing, again, has been spoken about by several different companies here, which is the idea of using genome editing. And the fundamental process here is you engineer a protein or a pair of proteins or an RNA-based system that recognizes a specific site in the genome. That engineered nuclease makes a break. And at that point, the break then activates the cell's repair machinery, and it can be repaired in one of 2 ways, either by non-homologous end joining, in which the ends are functionally stitched back together. And if there's repeated cutting and repair, you end up with small insertions and deletions at the site of the break. So it allows you to inactivate genetic elements. If you provide a piece of donor DNA at the same time that you create the break, you can actually trick the cell into using homologous recombination, or use its homologous recombination to use the donor DNA to insert the precise sequence at the site of the break. That can be a single nucleotide change. That can be a large cassette of genes. And just to make a point, since DNA is our target here, essentially every genetic element is druggable. There's no such thing as an undruggable sequence in the DNA. And I say that quite confidently because we have targeted nucleases to regions of the genome that are heterochromatinized. So while chromatin certainly has an effect, it is not deterministic. So I'm going to now focus on SCIDX1. This is caused by mutations in a gene called the interleukin-2 receptor gamma chain gene. It's part of the receptor complex for a number of different cytokines. The important ones are IL-7 and IL-15. So without signaling through IL-7, you do not get T cells, and without signaling through IL-15, you don't get NK cells. It's on the X chromosome, which is why this is a boy disease. And as I showed earlier, the mutations are scattered throughout the gene. One of the important reasons we've picked this rare disease, ultra-rare disease, so probably 20 cases in the U.S. per year, an incidence of 1 in 200,000 live births, is that there are a small number of anecdotal cases of boys who genetically had the mutation but developed an immune system. And when you go and look at those immune cells, what you find is that they had a somatic reversion in a precursor cell, perhaps a single precursor cell, which tells us that we don't need to correct a lot of cells here to have a beneficial effect. The current treatment for this, standard of care for this, is allogeneic bone marrow transplantation, and depending on the source of the donor, the outcomes are variable, can vary between being excellent with a 95% cure rate to less excellent, down in the 60-70% range. And this disease has been a target for viral-based gene therapy, and as many of you know, in the first generation of these trials, 25% of the patients developed a leukemia from the insertional activation of a proto-oncogene. Using second-generation viral therapies with safer vectors, so far that has not shown itself, fortunately. So one of the challenges when you're thinking about genome editing is, as I said, mutations that cause this disease are scattered throughout the gene. And with really the advance in nuclease technology, one might think that you could design a specific nuclease for every mutation in this gene. But what we really wanted to do was design a single platform that could correct all or most of these mutations. And the strategy that we're developing is the idea of engineering nucleases that hit right near the stop codon and then targeting the insertion of the wild-type cDNA into the endogenous locus. So you use endogenous regulatory elements to drive the wild-type gene, and in this way, for this disease, that strategy would correct 98 to 99% of the mutations that cause the disease. So we first made TALENs that targeted this exon 1 region, and we showed using an assay that measures the frequency that you create insertions and deletions at the site that our TALENs are active, where we could generate allele modifications of 33 to 40%. And what was reassuring to us with these TALENs is that modification frequency stayed constant over time, in contrast to when we use ZFNs to modify different genes. What we find is that we get high modification frequencies early, but we lose those modified cells over time, suggesting that there's a degree of toxicity with the zinc finger nucleases. So then we've taken this into CD34 positive cells. We've used a donor vector here in which we have a wild-type cDNA followed by the an expression cassette for GFP, showing that we can insert, as I said, a couple genes. And what we find is in our best case scenario, we can get somewhere between 2 and 3% of our CD34 positive hematopoietic stem and progenitor cells corrected. And on average, this is about 1.5%. So probably enough for this disease, but we also then wanted to go on as the CRISPR system got developed. We wanted to compare the TALEN nuclease system to the CRISPR system. So we made CRISPRs and TALEN. We made a guide RNAs that targeted essentially the same site as the TALENs we had previously engineered. And in cell lines, we actually saw that the CRISPR system worked 50% better than the TALEN system. But when we moved this standard DNA system into CD34 cells, what we found is that the CRISPR system was tenfold less effective. There's actually an error bar there. The efficiency of modification went from 1.4% to 0.12%. So there was something about the system that wasn't working. So in collaboration with Agilent Research Laboratories, we had the hypothesis that what we needed to do was to deliver the guide RNAs as synthetic guide RNAs into these cells rather than relying on the expression of the guide RNA from a DNA plasmid. And so and that was recently published in Nature Biotechnology. And in the interest of time, I'll sort of go to the meat of the matter, which is that using guide RNAs that were synthesized, so fully synthesized, with modifications at the terminal 3 base pairs on each end to make the guide RNAs resistant to nuclease degradation, what we found is that the more the guide RNAs were made resistant to nuclease degradation, we could see high frequencies of modification both at the common gamma chain gene and at the human beta globin gene. If we did not have the modifications or we delivered the guide RNAs as DNA molecules, we saw again the activity went to 0. So now we have a way of modifying using the CRISPR system. Now the second aspect to getting gene correction via homologous recombination is we have to deliver the donor DNA. And what I haven't shown you, but what we and others have shown is that when you deliver DNA into cells like hematopoietic stem and progenitor cells, you activate a type 2 interferon response, a toxic type 2 interferon response, which makes sense. Foreign DNA gets sensed as an invading nucleic acid, and you activate the innate immune response. So we needed to develop a way of delivering without activating that response. In prior work in my lab and in other people's lab, we have shown that you can use AAV to deliver the donor vector into cell lines. And so then what we did was designed a donor vector that was serotyped in AAV6, so one of the specific AAV serotypes. And what we showed is that when we delivered the donor, which is expressing a GFP molecule, without a nuclease, we see a small number of cells that are green initially, but over time, as the cells are cultured, those green cells disappear because the AAV episomal vector gets diluted. But if we co-deliver that either with TALENs or the CRISPR-Cas9 system, we see stable integration of the expression cassette such that with the TALENs, approximately 5 to 8% of the cells stay GFP positive, and with the CRISPRs, we see about 12% of the cells remaining GFP positive. That was delivering the CRISPR-Cas9 system as a full RNA system. We then, what you can do also is deliver the CRISPR-Cas9 system as a ribonucleoprotein complex, where you complex the guide RNA with the Cas9 protein and then deliver that into cells via electroporation. And what we found is the modification frequency using the RMP versus a full mRNA system is slightly lower, in this case of 13% to say about 17%. But what we found is by delivering with RMPs, we got higher viability and the cells do better. So it's a less toxic approach. Importantly, not only is it less toxic from a cell processing standpoint, we get higher specificity in that we see less insertions and deletions at a known off-target site for the guide RNA we were using. So we get higher specificity as well by delivering the CRISPR system as an RMP. So we're working towards scaling this up, and I'll get to it. So one of the important things that has been mentioned by all the people who are interested in genome editing is, of course, we have to assess safety. And I would just make 2 points. This is that there's no single preclinical or even set of preclinical assays that can tell us what is going to be an adverse event. And I like this quote from George Box, who on his wiki page is called one of the greatest statistical minds of the 20th century, so that may have been put in by his wife. But he has a quote that says, essentially, all models are wrong, but some are useful. And so the point is that we're going to have to use a series of model systems to assess toxicity and safety, and from this take our best guess about what's the safest formulation to use clinically. And there's a bunch of assays we might use, and I'd be happy to talk about the pros and cons of these later. So in the end, what we need to do is manufacture an autologous gene-corrected cell product. The idea would be we would harvest hematopoietic stem and progenitor cells from a patient, and then there are cell doses that we know we would have to get to that are both based on allogeneic transplantation and autologous gene therapy-based approaches. We would then bring that into a processing lab in which we would have to deliver a GMP-grade nuclease, a donor vector that's GMP-grade AAV6 using a GMP-grade delivery device, an electroporation device, and then we would transplant those autologous corrected cells back. And again, based on disease-specific numbers and autologous cell transplantation numbers, we have benchmarks that we know that we need to hit. This will all be done in our new laboratory of cell and gene medicine that's now being headed by Dr. David DeGiusto, who we stole from City of Hope. And I'd like to thank you for your attention. I'd particularly like to thank Rasmus Back, Danny Deaver, and I.L. Hendel, 3 very talented postdocs in the lab. Our excellent collaboration with Agilent Technologies, which I think shows that we are a good partner to people. And I would like to thank CIRM, who is funding our preclinical development of the SCID-X1 project. I will note that this grant required partnership, and my partner was actually a philanthropic donor. And so, again, we're very interested in partners. And with that, I will say that we are building a program in definitive and curative medicine at Stanford. And again, that program is looking for partners as well. And I think that's my last slide. And so I'd be happy to take questions or comments at the break. Thank you very much. And I had 5 seconds to spare.